We're live with our Microbiology Journal Club, so it is May 5th, and this is actually Providence College's last day of classes. So I got done with my class this morning, and so now I get to relax until we give the final and do a journal club, so I'm very excited about that. So today we're going to be talking about this paper, which is High Taxonomic Variability Despite Stable Functional Structure Across Microbial Communities. So we're going to be talking about bromeliad plants, which is a fun word to say. But before we get started, we'll do some quick introductions. So my name is Laura Williams. I'm an assistant professor at Providence College in Providence, Rhode Island. And my research group does microbiome work. So this is a helpful paper for us to be thinking about microbiomes and kind of the ecological side of microbiomes, which I think is a good perspective to bring into it. And then next to me in the little series of panels is Nicole, so I will let Nicole introduce herself.
Hi everyone, I'm Nicole Succio, and I am a postdoctoral fellow at the University of Northern British Columbia in Prince George, British Columbia, Canada. I have been also working with amplicon sequencing data that would be used in microbiome studies, but in my case, we've been focusing a lot on forest soil systems and looking at how soil handling in the short term affects community composition when we attempt to reassemble trenched areas. So this paper is also pretty interesting because of thinking about how communities shape themselves around detrital inputs, which will become clearer when we talk about the actual tank part of the bromeliad. And that's all I've got for now.
Yes, I like that they're called tanks. That's very exciting to me. It's a great term. Kat, would you like to introduce yourself?
Hi, I'm Kat Milligan-Myrie. I'm an assistant professor at the University of Alaska Anchorage, and I study host microbe interactions in sticklebacks, so we can determine how the host genetic background contributes to the relationship between microbes and their host. I'm excited about this paper because I did my postdoc in Karen Gillerman's lab, and we spent a lot of time thinking about how communities are assembled, and whether it was neutral processes or something else that was driving microbial community structure. I wasn't involved directly in those studies, but I heard a lot about them, and so I'm excited to think about this in the context of plants.
Excellent, thank you very much. And I'm going to have Joel introduce himself next, but before I do that, I'm going to quickly remind Kendra that she might want to mute herself.
Oh,
if she's
sorry, I thought I was.
That's all right. So, and then we'll have Joel introduce himself.
Hi, I'm Joel Groff. I'm an assistant professor at Montana Tech in Butte, America, and I mostly study human pathogens, but I've been interested in microbiome research in the human body. So when I read the abstract of this paper, I thought they were going to be comparing taxonomies versus gene expression, and it's a little bit different than that. So, but it's along the lines of what they've seen with microbiota and intestine. But anyway, I do not have much background in this, but I actually only recently learned about something called permaculture, which I've heard the term for years and years, but only recently decided to figure out what it might be. And so soil microbiota has gotten much more interesting to me since I learned about that. Anyway, that's enough for me for now. Thanks.
Thanks, Joel. And then last but not least, Kendra, would you like to introduce yourself?
Hi. Sorry, I thought I had my thing on. Kat and Joel, it's nice to see you. Hi. And Nicole and Laura always. But so I'm Kendra Moss. I am a facility scientist at the University of Connecticut. I run a core facility where I help other people do sequencing. So mostly I do amplicon sequencing, so lots and lots of microbiome. My background is microbial ecology, and I particularly love this paper, having read it today, because I am trying to finally finish one part of my PhD research, which was on hot springs and the bacteria that live in them and the geochemistry. And so this paper is oddly perfect for that, which when I read the abstract briefly, I didn't think it was, because I thought it was all going to be metagenomics and functional genes. But yay!
Yeah, I think I have to admit that when I read the abstract, I was like, I'm not entirely sure that I know what they're going to be talking about from the abstract. But the paper itself is kind of launches into some interesting things. I think it's a paper that would, if you're a microbiologist and you're reading this paper, you'd be like, okay, if you're an ecologist who's getting frustrated about the microbiome not bringing ecology into the picture, you might be a little happier with this paper because they are trying to kind of think about environmental conditions and what is actually shaping the microbiome of their particular study organism. So maybe the way we'll start this is to talk about what exactly are they looking at. So they are looking at these bromeliad plants. I am not a botanist to any degree, so I'm not going to claim to know anything about this, but let me do a quick, I'll just show, if I'll share my screen here, and I will show a little picture. There we go. So that is your bromeliad, an example of one of the bromeliad species that they used in this study. So I think they only looked at, there are many different types of bromeliads, but I think they only looked at this one species. And they've got in the center there, they've got this, this well or this cavity that they call a tank. And it's going to accumulate rainwater, it's going to accumulate detritus. I'm assuming you could get things like little dead insects in there and so forth. So it's going to kind of accumulate this organic matter, and it's going to accumulate, my guess is that depending on the weather, the level of moisture is going to be changing depending on when they've recently had rain and so forth. So one of the things I was wondering is, do we think this is supposed to be kind of like soil, or is this just going to be its own completely unique kind of material? That's a question that they don't really address, but I was just curious if anybody either had any experience or any thoughts about that.
I'm going to jump in here and say that I think it could be both, because I hadn't really thought about the whole arthropods either incidentally or intentionally coming to die in the tank, but things kind of do that in soil too. So the one thing that's interesting about this is you've got these two ideas. One is that the bromeliad tank is a cool system in terms of site occupancy. So the likelihood of things that fall in coming out not very high unless it's really windy or they get sloshed or something. So it means that you have biotic processes, metabolic processes, and environmental shaping that can happen around inputs that are going to have an appreciable residency time in the tank area. The second part about this is thinking about substrate availability and substrate quality, which is addressed to a certain extent in the paper with some of the measurements they take. But you're ending up with these chemical assemblages of different things in different states of decay. So there could be insect carapaces, there could be dirt that's kicked in, there could be litter material that falls in. So we have substrates of different reactivities that will be useful to different parts of the microbial communities that are also inoculating that space that come in with detrital or aqueous input at the same time. So I think there's some good bridges there in terms of the resource context being as heterogeneous as a soil could be, or as heterogeneous as a water compartment that has a high VOC or carbon input.
Just to play devil's advocate, it seems like these tanks are pretty much isolated, and so they're a little bit not as free-flowing as you'd expect in the soil, moving around from one area to the soil to another. So it's maybe an intermediate between the two.
Yeah, I think that they are just a really great enclosed ecosystem. Like, you know, this is an entire watershed all by itself for microbes, is the way that I read it.
Yeah, that makes sense to me. And I think given some of the questions that they're asking, so what they're really trying to get at with this is, what are the processes that shape the microbial community composition in terms of who's there, what can they do, functional capacity, and then abundances. And they're asking, they ask questions about dispersal, spatial relationships. And so I certainly see how it helps that they've got kind of, if you tried to do this in soil, like Joel pointed out, you'd have a lot more flow or exchange or so forth. If you've got these plants, you know that like that one tank of one plant is kind of a distinct, it's almost like you took a graduated cylinder and you stuck it out in the middle of your field site and you're like, okay, there's cylinder one. Like that's what your bromeliad plant is. So I could see that being useful for what they're asking.
One of the things I found interesting about the system is that it's not like a gut that has microbes coming in and microbes going out, or a water system or a soil system where you have an exchange. One of the things that somebody pointed out earlier is that it seems to be an enclosed system, but that also means that the microbes don't have anywhere to go. And so in their sampling, they're doing DNA analysis, and that's also going to include all the things that fell in there and then died. And so I was a little curious about how they are going to handle that. How are they going to determine whether or not the things that they see there are actually providing some sort of function to the community, or if they're just transient, it's hard to think about a transient microbial member in this particular community because they don't have a way to leave. It's not like the gut where they can come in and be excreted and then they're gone. In this particular system, they come in, and unless they have a way to actually move up the leaf and get out again, they're kind of stuck there. So it's a little different than the systems I'm used to thinking about. I hadn't thought about the fact that it's like a graduated cylinder where they come in and then they're there. There's not really a good way for them to leave. So it's going to be interesting. Yeah.
Yeah, and I think that's a really good point because they don't, they focus on DNA analysis. So the DNA that they're going to recover, and then they do a lot of like physicochemical testing to try to kind of determine things like pH and so forth. But Kat, you've made a good point that nothing that they, unless I missed something, nothing that they did actually addresses viability. So they didn't do any culturing or so forth. And, we actually had, Pat Schloss was nice enough to Skype with my microbiology class earlier this, I think it was last week. And that's one of the things he said about microbiome studies. He said, like, look, a lot of the stuff that people claim to find in, low biomass samples, like the placental microbiome, you could culture those. So you get so obsessed with like sequencing and data analysis and DNA that you kind of forget that, you could go into a lab and try to see what do you get that comes out on a plate. So that's not to say this isn't valuable, but that's a piece that I think was worth thinking about.
Yeah. In defense of never culturing anything, which other people can culture, it's just not me. You should only be having a few random cells fall in at a time. So if you can measure a particular species, it probably was able to live at least for a while. It might not be currently alive, but it was at least there. This is something that for my hot spring stuff we worry about, because you can get like a big chunk of, say, bison dung fall into a hot spring, and you would have a lot of bacteria in that. So we could be measuring something that was never living there. These bromeliad tanks, it should all be airborne that's getting blown in. So that should be single cells rather than huge chunks. That's my belief. Yeah, except that they took the debris from the bottom of the little tanks, and so that's going to include, like she said, the insects that land in there and all the things that are in their gut and the dust, and and dust, okay, is probably going to be pretty small, but if you have like a chunk of leaf that falls from one of the trees up above, because it looked like from the picture that these are in kind of foresty areas, it's a big chunk of debris that's probably has a microbial community of its own that might contribute to the community that has nothing to do with, it could have something to do with the community itself, because it's going to be contributing members and it's going to interact, and it might change the community because it's falling into this tank. But when you're thinking about assembly and you're thinking about community structure, keeping track of which ones are alive and which ones are dead is kind of important for a system like this, where you might have big chunks of communities that come from elsewhere.
Yeah, I think those are all really good points to think about what's, because one of the things they get into is functional capacity and metabolic groups, which we'll talk about in a minute. And so certainly if you're if you're going to start classifying things on the basis of their metabolic capacity, your assumption is that they're performing that metabolic capacity, that it's playing a role in the community itself. So yeah, I think those are all interesting constraints or things to think about in terms of the structure of the community that they're looking at.
Okay, so I thought of another something though. In defense of what Kendra is saying, now that I think about it a little more, as the cells are dying, their DNA is going to be released, and that'll be broken down by other microbes that'll be present in DNases and all those fun things. So, you know, maybe it doesn't, it's hard to say whether or not it's going to play a big deal in this. I guess it depends on like how long ago did that insect land in your little tank. So yeah, just something to keep in mind as we're looking through this.
Yeah, and a larger point is that it just kind of gets at how complex this is to try to kind of account for all of these variables, which they account for quite a few, or they make a good effort to.
So my thought on this is, as I was reading this paper, I went into it thinking that they're going to do transcriptomics and compare that to their 16S, and because I think that's what they've done in the microbiome. But you know, transcriptomics might be a better proxy for whether something's living or not and actively metabolizing things, just because of its short half-life and that sort of thing. So I was just trying to decide which is better, RNA-seq or this pick out a few characteristic genes and look for those. And I'd say this discussion right now is kind of pushing me towards RNA-seq.
Probably, except lots of environments you don't have the reference. So RNA-seq, you get a huge pile of, you know, gene 1, gene 2, gene 3, but you don't have a reference to know, to be able to ID those very well. I think that's probably why they went targeted gene.
I'm just going to jump in and say that with some of the work, I did do a small module of attempting to do my amplicon sequencing from cDNA that came from soil-derived RNA, and very similar to what Cornelia Smala has observed when she has attempted to do the same thing in her soil systems. And yes, this is a very soil-centric perspective, is that overall community compositions do not tend to differ that much. But I mean, we're also dealing with a lot more taxonomic hyperdiversity than might be potentially within a bromeliad or a system where you've got a lot more soluble substrate flux or changes in conditions for that microbe that might actually change fluxes with a bit more sensitivity. I mean, when you're doing it in a non-fluidic system, you have to expect to be a little disappointed, which I kind of was, but it was also good to see it. So that's my perspective on trying to do it from the RNA end of things.
Yeah, that's a good point. So yeah, that's an interesting discussion about the constraints that they're dealing with here. So thanks everybody for that. That's very enlightening. So what I think we'll do now is we'll kind of move into, I think the best thing to do before we launch into the data is to do a little bit of setup about because we've been talking a little bit about the data that they are using. So they did go after genomic DNA, and they did two things. So they are interested in both taxonomic composition, and they're also interested in kind of functional capacity. So following up on the what we've seen with, they mentioned bioreactors, which I don't know as much about, but they also mention what we've noticed with the human microbiome, which is if you're looking for a core set of OTUs or a core set of microbial taxa, you don't really find that in the human microbiome. But if you look for a core set of metabolic capacities or a core set of functions, you do tend to find more stability or more shared functions than you do more shared taxa. So they're starting to look at that in this system to try to understand, well, okay, if you've got shared functional capacity, we want to understand what, if we start to break those functions into categories pretty broadly, we're interested in how do you get that functional capacity? Do you get it from lots of different OTUs, lots of different taxa, or you start to kind of see the same taxa showing up in a functional group? So that's kind of what I gathered was the thrust of what they're doing here, is trying to kind of look at community assembly. How do you get to this functional capacity? So to do that, they're looking at both 16S, and they're also looking at shotgun data. So they did 16S data and they did shotgun metagenomics. And I just want to, because I thought this was, they were very upfront about this, so I appreciate that they talked about this. In the methods, they have to kind of figure, so taking your 16S data and classifying it in terms of its taxonomic membership is something that everybody does this with their 16S data. They take a Silva database, that's pretty reasonable. But then what they talk about is functional annotation of prokaryotic taxa. So what they're going to do is they're going to take the taxonomic classification based off 16S, and they're going to classify that OTU into a functional group. So they list 9 functional groups that they decided to focus on. So these are broad categories, things like aerobic chemoheterotrophy, cellular lysis, fermentation, methanogenesis, methylotrophy, nitrogen respiration, sulfate respiration, photoautotrophy, uriolysis. So they've got these 9 categories. And what they did was they said, so an OTU or a taxon was assigned to one of those 9 functional categories if all cultured species within the taxon are known to exhibit that function. So they had to go, I mean, it sounds to me like they had to go back through the literature. They had to look at what people have reported for the cultured relatives of this, this OTU that they've got. And they had to say, okay, on the basis of everybody reporting that this particular thing will digest, the cellular lysis, or it ferments this, we're going to put our OTU into that functional category. And they're upfront with saying clades with no cultured representatives were not functionally annotated. So I'm guessing that's a pretty good proportion of what they're looking at here, is they're going to be pulling out OTUs where they're not going to find a close relative that's been cultured and well characterized. So a big chunk of their data, they just have to kind of drop out of this. Later on in the paper, they kind of say the conclusions that they're coming to probably are not going to be affected by that. Like if you included those OTUs, it would probably support what they're saying rather than contradict it. But I was pleased that they kind of walked through this in very clear language to say, we did this, but we do understand that these are the limitations. So let's see, the other thing that I wanted to say was you can get an OTU that's classified into 2 different categories, functional, so it can be classified into more than one, which I'd written that in the intro and they answered it in the methods, so I was happy with that. And they talk about why they had to do this, which is, it's really hard to taxonomically bin shotgun sequences. So they can't go the other way, basically. So they need to take their taxonomic classification that they're confident about and try to assign it a function. Because if you take your shotgun data and try to assign it to a taxonomic classification, forget it. Like I think a few years ago, Nick Lohman, bioinformatician, showed this, where he took an E. coli genome and he shredded it into like 250, maybe less than 300 base pair segments. And he sent that through a classifier, like a metagenome classifier. And it came back mostly E. coli, but then like Klebsiella, Citrobacter. And he's like, this is not Klebsiella, it's Citrobacter, So it's hard to taxonomically classify your shotgun reads. So they went the other way with it. They took their taxonomic classifications and assigned them to functions.
Yeah, which is really interesting because I'm so skeptical of assigning function based on taxonomy. I am going to try their tool. So they have this tool out there. I'm going to try it with my Yellowstone stuff since I'm working on it right now, and just see what it gives me and if it makes any sense. Although what I'm going to, ground truth that against, I don't know, because it seems like there's been so much work on. And I have been skeptical of it, but there's been so much work of people doing these shotgun libraries and then comparing their taxonomy from their shotgun library to their 16S libraries. And that actually has been one of my main, Nicole has heard me rant about this, like one of my main critiques of shotgun libraries is it seems like people are just using them to say, look, my 16S works or not. And I'm like, that's a lot of money to not do anything other than say, here's my taxa. So it's interesting that they said that, and I wonder how much pushback they got from reviewers in saying, all this stuff that people have been doing for a decade is nonsense.
I wondered if anybody, yeah, no, go Nicole. I was wondering if anybody had thoughts on that.
Yeah, it's my 2-second half-baked biochemist soapboxing a rant, and that's just that the literature and start thinking about whether this makes sense based on biogeochemical cycles that have been documented. Like, you can't, these automated annotations aren't going to save you for thinking about whether this is reflecting nutrient fluxes that have been identified. And I mean, this is why, again, I'm very thankful with the fungi stuff that a lot of the functional guild classifications have worked out, but it's still, you know, there's still things that you have to take with a grain of salt with that. But like, this is where sort of moving laterally outside of a sequence set of data, there isn't going to be a magical intuitive thing that pulls out once you have all that data in hand. And I will say that the one thing with the FAPROTAX application that was developed and reported on in this paper is it's conservative in the sense that it's picking singular metabolic niches. It's not attempting to like predict entire metabolic pathways, which has been a feature of another tool that has been developed to parse 16S data. So I think I can appreciate the disclaimers that they put on here, because manual curation is an attribute of many other ontologies that have attempted to be used with metagenomic or 16S data. I mean, SEED is about bringing together a consortium of supposed subject matter experts. So you're always going to have gaps, expertise in that case. And in this case, this is also kind of neat because it's at least an attempt to try and merge chemical taxonomy, which has been sort of a foothold in classical bacteriology for a long time, with linking it to 16S. So going back and attempting to bring those two aspects of classification together, well, you know, all aspects of classification are inherently going to run up against being comprehensive. So do your lit reviews laterally and think about your results, and also, again, be as honest as they have been, because this paper's an awesome playbook for justifying what they did and what it can't do.
Yeah, I do appreciate that. I appreciate that. I mean, and I liked it because I felt like it was really clearly written, right? So I feel like it very clearly walked through, like, here's our rationale. We understand this limitation. We understand this limitation. And I like it. I like that they're upfront about that. That's really good. So that, I wanted to make sure we were all aware of that before we moved on, because they do rely very heavily on their functional classifications. So does anybody have any other points you wanted to raise about that particular aspect of the work before we go to figure 2? Okay, I'm gonna take that as a no. So, all right, so one of the first things they wanted to do is look at, as figure 2 says, taxonomic and functional community structure. So I'm gonna pop up the paper, so, which means I don't have to look at myself anymore. That's also very exciting for me. So let me pop up the paper, and okay. Oh, can everybody see that? All right, so the top of the paper is panel A and panel B. There they are looking at the, so that's relative abundances of proxy genes in prokaryotic metagenomic sequences. So genes are grouped by function with one color per gene group and one color per bromeliad. So what they're saying here is they're actually gonna argue that this is, they've got, let's see, so metabolic functional structure in terms of relative abundances of proxy genes was similar between all bromeliads. So my understanding is that they're pulling this from the environmental shotgun sequencing data. So this top row is not their 16S data. This top row is their shotgun data, where they're trying to kind of figure out what's the functional capacity of these communities. If that sounds off, somebody just wave wildly. And so what they're noticing here is across these bromeliads, which are the columns, they've got 22 of them, and they're seeing a lot of stability in these functional categories that they're showing you on the very right of this top row. So fermentation, oxygen respiration, carbon fixation. You got some variability, but overall they're arguing that this demonstrates functional stability. And then I noticed that they've got this the little gray thing that's in between A and B. They're actually expanding their rare gene groups, their rare functional gene groups, so you can get a better look at the scale for those. So those are things like there's a little bit of like methylotrophy. That's the little red at the very bottom of 2B. And so they're going to say, okay, we've got stability in our functional community structure, and then we want to understand within these functional categories who is doing this stuff. And so then they go down to the rest of the panels and they start grouping some of the categories together so that they can get their 9. So the top of each panel tells you what that category is. So 2C is the fermenters. 2D is a combination of 3 of the functional categories that they were talking about before. So they've got these 9 functional categories, and this data is their OTU levels. So this data is their 16S data. And they're looking at, for each one of these bromeliads, who is doing the fermentation, who is a methanogen, and so forth. And so here they're basically saying this is really highly variable. You do not see a lot of core across bromeliads. They're making the argument that you don't see really a core set of microbial species or microbial taxa who are performing these. What do you guys think about this? Is this, I mean, my understanding is this is pretty much from, they mentioned bioreactors, but this is kind of, I don't think this is too surprising to anybody, but does anybody have thoughts on this?
So I'm going to go here, and I'm going to say that for me, I'm not incredibly surprised from this, because when you also think about this bromeliad tank system, as Kat pointed out, it's basically a pitfall trap system for whatever happens to end up in the tank. So it's bringing, again, a subsample of whatever's in the environment that would be associated with a detrital pool into that material. So you're starting with a certain initial community, and also there's going to be some, again, temporal and perhaps bioavailability limited conversions of that matter, which are probably shaping that community as it is. So I think there's 2 very large coefficients of heterogeneity to, you know, the community configuration as it will be, even though it makes sense to me that you have stability of the metabolic component, because again, you're going to have some type of mechanism of control that's creating this assemblage of organisms that operate at the kinetic paces they do and operate with the timing of accessing substrates that they do. So that's really hand-wavy, and I don't know if it made sense, but for me, this is consistent with the sort of biological profile I'd expect to get out of this sort of data with this experimental system.
Yeah, I mean, I agree with you. I think that that's, and I think this leads into, I'm going to move a little bit to lead into, hopefully this doesn't make anybody ill when I do this. Sorry if this is making people paper sick. But here, so figure 3, they kind of tie figure 3 in to help explain figure 2. So figure 3 is the functional redundancy in the regional OTU pool. So I kind of understood, I think the regional OTU pool is basically, I think that's meant to be like everybody. I think they just combined all 22 samples to try to understand this is our overall representation of who is around in this plot in general. And so we're going to look at those as a kind of combined pool. And what they've done here is they've got their 9 functional categories represented as rows. And then they have OTUs represented as columns. So everywhere you see a little blue hash, that means that that particular OTU in that column got classified into the functional group. So up at the top, there are a lot of OTUs that got classified into aerobic chemoheterotrophy. And if we go back up, again, I'm just don't want to make anyone sick, but if we go back up, that's figure 2D. And 2D is like this patchwork quilt of all kinds of different, taxa. So the figure 3 plus 2D is kind of not surprising. So it looks like you have a lot of OTUs who have this functional capacity, and that means that this role, this niche is getting filled by whoever happens to fall in there that can then fit into that particular broader biochemical pathway. And I think they've shown that too. Like their next one is fermentation, where they've got quite a few OTUs that are classified as fermenters. And if you go back up, your fermenters are also another one where you've got this real patchwork quilt type effect for their 16S OTU data. Kendra, did you have a thought on this?
So I generally love stacked bar plots, but I honestly did not look at figure 2 very long because it made me sad. Like they're presenting so much data here and it's so uninterpretable. I really wish that, oh, sorry, I'm trying to scroll your screen. I really wish that figure 3, they had another bar that showed taxonomic affiliation of each OTU. Like, I feel like that would have made these figures so much more useful. And I want to believe what they're telling me. It's just like they're not connecting those dots between these figures. And I find figure 2 just colors with no legend. Makes me crazy.
Figure two is pretty intense. It's true.
Yeah, like you can't use colors if you're not going to have a legend that explains what they are. And they don't. Their legend is only for panels A and B. This is where I am sad by this paper, because I think it could have been, I think figure 3 could have been the figure of the paper to really explain that you have a wide range of OTUs doing all of these different functions. And it makes me sad. And I'm sad that they don't have that in their supplementary either.
What do you think would have been, if you wanted to do away with the stacked bar charts in 2 entirely, what would there be, because this is always something I'm thinking about. I'm always trying to think about data visualization because for whatever reason, it's not an intuitive thing for me, and I have to actually kind of think about it and work on it and try to get better. So I'm kind of curious, what would have been, would it have been more useful to show a plot of a metric, like even if it's just richness or some type of alpha diversity index? Would that have been more useful for each of these?
I absolutely think that an alpha diversity index would have been so much more useful there.
Mm-hmm.
Because frankly, that you can't, like if you look at C or D, there's all those colors, but like I can't count how many there are. There's just a lot. So yeah. And I've done some box or some heat maps. I don't love heat maps generally, but I think their use of heat map in figure 3 is useful. But I think it would have been so much more useful if they had included that taxonomic information in that same figure, so you could look at it and say it's there. But I love thinking about data visualization. So I think a metric there would have been better.
Yeah. Joel, go ahead.
Yeah, it's been a year and a half since I've regularly sat through lab meetings where we talked about microbiome studies. And I remember alpha diversity and beta diversity, but just vaguely. Could someone remind me of those?
I'm the self-identified ecologist. Alpha diversity is the diversity within a single sample. Beta diversity is diversity between samples. And generally the way that we calculate that is between pairs of samples. So if you think of, like on a map, you have the distance between Butte and Missoula, and then you have the distance between Butte and Bozeman, and then you have the distance between Butte and Yellowstone, because everything goes to Yellowstone, right? So you get these lower triangular matrices where it's pairwise differences between samples. And then to get what they're showing in figure 3, I think you could actually talk about that being gamma diversity, which is the diversity not just within an ecosystem, but across these ecosystems. So they could argue that this is a gamma diversity situation.
And I think they actually do make a mention of regional gamma diversity in the text, yeah.
Which is something that microbial ecologists don't talk about very much, because it's really hard to define what's an ecosystem in it for a microbe.
Sure. Yeah. So for I think that was a great explanation. I'll just follow up on the alpha diversity. It's something we're kind of doing with our lab too. the alpha diversity is going to account for, so depending on what you choose to report, if you would report something like a inverse Simpson index. I was just talking to one of my students about this morning. So though you can get a metric that should account for what's called richness, which is just the straight up number of observed OTUs. But you can also try to account for evenness, which is that you could have a sample that has 20 OTUs, but one of those OTUs is like 99% of your reads and everybody else is just this tiny fraction. You could have another sample that's 20 OTUs, but they're all evenly represented. So I think what we're kind of coming to is maybe it would have been useful to see one of those metrics reported that tries to account for what the stacked bar chart is showing. Because the stacked bar chart is attempting to show both the total number and then also differences in abundance. And it just visually, like Kendra pointed out, it gets the point across that there's a lot there, but maybe reporting an alpha diversity, maybe there's a suitable alpha diversity index out there that could have done that too.
And so I'm a huge fan of plotting a couple of different alpha diversity measures. I'm also a huge fan of plotting a couple of different beta diversity measures because they all emphasize different aspects of the community. So if you plotted inverse Simpson versus an evenness metric or just species abundance, like just make a scatter plot like that, that could have told you something about how these communities were organized, or how these different metabolisms are organized. Like that could have been an interesting thing to plot. Sometimes it's not, but sometimes you can see real differences in your communities that you wouldn't necessarily notice looking at just one metric.
Yeah, that's a good point too. Any other questions about 2 and 3? So I think the big thing coming out of 2 is, and supported by 3, is that they've got stability in their functional community structure that is provided by a whole wide range of microbial taxa. So taxonomically, they don't have a core set. They have a lot of different taxa that are combining in different ways in different individual plants to give you these stable functional categories. So, all right, so then, and there isn't a figure for this, so I will stop my screen sharing. All right, so then they get into a lot of text about trying to explain what's going on. So they talk a little bit about, oh man, the null models on this. Boy, I read the methods and I was like, oh wow. Okay, all right. So, and they have a lot of, I liked this. I liked that they're trying to kind of define what is your null model. Like what, if you're gonna make a claim about testing these different processes and what process is shaping this community composition within these functional categories, I like that they're like, we're gonna try to define a null model and see what we can come up with. So the first one that they kind of talk about is OTU co-occurrence. And as I mentioned, there's not a figure that goes with this. So this is all stuff that's coming from the text. So they got this C-score that they're going to calculate. And so they're going to try to determine when the community is being assembled, do they see what they keep calling segregation of OTUs? So do they see OTUs that are, you could interpret this in a couple ways, OTUs that are always occurring together in a sample, or if you find one OTU, you never find this other OTU, and vice versa. So they're trying to kind of figure out about associations between OTUs as the community is being assembled. And so they make the kind of null hypothesis statement that random colonization of bromeliads by independent OTUs would result in negligible association between OTUs. That makes sense. So then they're going to say, okay, we're going to look at just a presence-absence kind of, is it there or is it not there? And when they run their C-scores, they say for 6 out of their 9 functional groups, the OTUs are significantly segregated. So they're seeing that the OTUs co-occur less frequently than they would expect by chance if it was random. So then the thing I appreciated about this is they start to kind of walk through potential explanations and they try to kind of knock them out one by one. So they say, all right, so failure to detect significant co-occurrence patterns, maybe that reflects random assembly processes, but it could also be due to the fact that we reduced abundance data to a binary of present-absent. So what we're going to do now is we're going to redo this with a null model that's based on abundance. so that we're accounting for that one OTU may be very abundant and one OTU may be rare. So now we're going to do it that way. And so they do that. They come up with an abundance-based metric, an MA score, and they again we found that all functional groups exhibited an MA score that was significantly lower than expected, indicating a substantial non-random segregation between samples. So again, they're backing up what they found before, which is that it's not randomly assembled. It appears that there's some kind of association between OTUs. So they talked a little bit about what are some other ways this could occur. But I think in the end what they're finding is that they have a reasoning for why they don't think that it's spatially limited dispersal. So what they're saying here is their kind of conclusion from all of this is the non-random segregation patterns detected here probably reflect mutual exclusion between OTUs that was potentially caused by environmental filtering or biotic interactions. So they're kind of now starting to try to interpret this and pinpoint processes that they think are shaping the communities in terms of who's there. Thoughts on that? Everyone's like, no, I have no thoughts on that.
So my big thing with this is, again, it's coming back to this idea of what they directly sampled was the detrital material. And again, it's hard when you've started with a bunch of bromeliads, because this is a cross-section in time where they've taken 22 of them and taken just crap that happened to be in this bromeliad, which means that there isn't a temporal unification of the stages of whatever transformations are happening in that detritus at that one time. So I mean, I agree with the statement of biotic interaction, but what it means in terms of community successions that might be accompanying the type of chemical substrate quality that's in the detritus, as well as where it came from. This might just be arguing semantics of what spatially limited dispersal is, because also there's going to be compartmentalized heterogeneity of detritus anyways. Like there's going to be parts of it that are turning over and not uniformly distributed chemicals that are going to be throughout the whole thing. So it's an interesting way of looking at, I'm still thinking of this as inoculation events. And if people want to disagree with me, that's fine. But like that is really what my head is doing with interpreting this data set based on how the system's set up. So that's what I have to say about that.
I think that's an interesting point, but I also think that, I mean, this is, I have this discussion with people who study gut microbiomes all the time, because what we tend to do is we tend to compare the microbiome in the feces or the microbiome in the entire gut, and ignoring spatial differences, and not just along the gut, but also what does the mucosal community look like versus the lumen community versus the community that might be in between there versus some that are in big biofilm patches. But I think this is a problem that we have with microbial communities in general, is that we start out with this really broad brush of like what's there in total in the entire community. And then we lose track of the fact that there are going to be differences within that community. I like the biofilm community, the biofilm researchers, because what they tend to do is think about spatially who's in which part of the biofilm, and I think that we lose that when we look at these big broader pictures. And there's definitely the differences in the, the small differences in chemical structure and chemical composition between what's in the very center of that, the tank versus what's on the edge versus what's at the air liquid interface or what's at the very bottom that's got to be more anaerobic. But at some point you have to start somewhere. So I see this as like a beginning study to launch this model system as a way to study these. It's going to be interesting to figure out where they take this beyond this, because this is a great, this is what's here and this is the functional description, and we're going to tie it to chemistry. And I think what's going to be interesting next is how are they going to tie this into what these communities are actually doing for the plant and how to manipulate them and everything that comes after. The assembly thing is really interesting, but applying that to, I don't know, that's, yeah, I don't know where to go with the assembly part of their discussion.
Yeah, I think you're right. I think they've done a good setup in terms of, and there's a lot of things to recommend this particular system just because of it's got these isolated plants, and you can, but assembly, given all the stochasticity that we've talked about, is a tricky thing to try to kind of actually model.
Yeah.
There's a lot of variability.
Oh, sorry, Laura. I was going to say on the geographical distance and the environmental filtering aspect of it, it's not clear what the source is. So if you're looking at, like, I don't think that a tank is a source for another tank. I think that's really unlikely. So as much as I love that they're trying to do larger scale biogeography of microbes, these tanks aren't, like, trying to fit them just to a straight line geographic distance isn't necessarily, like, I'm not surprised they found nothing, because, you know, they're surrounded by their source. So it's not even like you can think of it in classical island ecology, because there's not a continent over here and the islands are here, right? Like, it's like it's islands within a continent. So I like that they tried. I really did. And I tried really hard with my Yellowstone stuff, and also had lines that looked like that.
Yeah, I thought I would pop it up there because you were talking about the geographic distance. So they tried, like you said, they tried. They're like, okay, so let's see if there's geographic distance on the x-axis, and then they've got Bray-Curtis dissimilarity on the y-axis. And so they're trying to see if they've got some relationships between microbiome community structure and geographic distance, and they don't really.
It's just not there. But I think this is also something that it's a little bit disappointing, because their whole paper is saying when you collapse them to these functional groups, they're all different. So then when they're looking at the communities by functional group, of course they're different. That's the whole point of their paper. So I guess maybe that's why they did that, is to say, see, look at, they're all different. But it would be interesting to see if there are any shared OTUs. And there definitely are some. There's the more abundant OTUs that seem to have a number. Well, I guess this isn't true. We don't know exactly how they're spatially segregated, so I can't say that. I'm sorry. I was thinking figure 3 was by sample, and it's not. It's by
OTU. Yeah.
Yeah. So this is, as much as sometimes I cringe when people do the core microbiome, because I feel like they're simplifying the community down to the weeds. But I think this could be a situation where it actually would have been useful to do this on whatever core they could find.
Mm-hmm.
And maybe they tried it and there was just no core worth looking at. Like, it could have been like 2% of the community is the core, and that's probably just weeds.
Mm-hmm. Yeah, and I think I wanted to kind of, on the heels of talking about the geographic distance, I also wanted to, with the little bit of time that we've got left, I didn't want to go completely skipping figure 4, but their whole goal here was to try to kind of start to think about the processes that are shaping this. And so they mention environmental filtering, and they mention kind of what are the roles of neutral processes versus non-neutral processes, biotic interactions, demographic shift, so forth. But they have a whole huge section in their methods is about just this painstaking characterization of all these environmental physicochemical conditions. And this is figure 4, right? So they've got detrital volume, dissolved organic chemistry, light intensity, methane, like on it goes, salinity. And in the end, it's kind of like what this is showing is the top of it with the 9 functional categories. You don't have a lot of the variance. This is how I read it. There is not a lot of the variance that's explained by any one of these particular environmental variables. So what's coming out of this to me is there is really no one thing that is like, boy, if you've got a pH of blank, then that's going to give you this, and it's going to give you that as it goes up a pH gradient. Like there's no one environmental condition that's jumping out as being, this is the organizing variable that kind of shapes what's going on with these microbiome communities.
Well, and my big takeaway from this, and I didn't read it super close, sorry, but it could just be that their entire range of pH is suitable to pretty much any of the microbes that fall into it. Like microbes are pretty tolerant, so it could just be that the whole range of habitats in these bromeliads, they're all the same species, so you would think that they're going to be fairly similar, and it could just be that it's better to think of them as 22 replicates that have, you know, naturally inoculated rather than some kind of environmental gradient. And that's what I got out of this, is that this isn't an environmental gradient.
Yeah, I think that's a reasonable way to look at this. Yeah. So, go ahead. No, please.
Yeah,
go
no,
ahead.
and I think to kind of look at this and also thinking about the explained variation that is on, I think, the last figure, the grayish, well, okay, it's gray to me because I didn't print the paper, and oh, it is gray. So like the last set of explanatory variance things is just the paper. I mean, it's a lot of like, there's a lot of work, and we're going to disappoint you because there's not a lot going on. But I think these ideas of using systems where things just fall in, and what they've done is really shown like the limited to non-explanatory translational value of assuming that you could use bromeliad detritus to sort of understand microbial composition that might be coming from the larger environment that the bromeliads are in. So I'm agreeing with Kendra's idea that you've got tanks as reasonably enclosed communities, and that's sort of an important thing for people to think about, is the linkages to the environmental variables they measured here aren't particularly tight or particularly powerful. And I mean, we turn to these methods of DNA and sort of coarser sampling designs in the hope that we don't have to do things that are spatially explicit, because on high throughput, or maybe from a monitoring standpoint, it would be nice to do that. But like, it's clear here that the explanatory power of what you're getting out of that in relation to a larger environment that you might want it to reflect doesn't pan out particularly in this study. So I think that's sort of the practical bent on it, and especially trying to use sort of the biogeographical numerical ecology that they apply to it as well.
Yeah, definitely. And I mean, I think so. We're coming up on 3 o'clock. I think, Nicole, you gave a really good kind of broad picture perspective on this. And so I'm not going to recap it because I think that was really good in its own right as a summary of kind of what you can do with this system, what its limitations are, and then what's it telling us about microbiome community assembly. Does anyone else have any kind of wrap-up thoughts you want to present for the paper?
So I'm thinking about how lucky I am that I'm not an environmental biologist, because I set up my experiments and I can control my parameters and all that. But could you grow some bromeliads in a greenhouse and just or even keep them separated or whatever, and then drop different things into them and then
like notobiotic bromeliads. That's a good idea.
Yeah. That's how I would go about trying to figure this stuff out instead of traipsing around Brazil. But anyway.
I think it's more fun to traipse around Brazil. I would, so if I were gonna make a notobiotic, I would do it in Brazil, because you have the right environmental conditions, right? You don't have to have the greenhouse and whatnot, and you could just take your little tank and then sterilize it, and then drop whatever you wanted into it, and then cover it with, you know, something to keep other debris from falling in, and you would still be in Brazil. So I mean, that's my favorite part of the system. Actually, I think there are things that would be that this system would be useful for, and I think just figuring out what the right questions is going to be the challenge for this particular system. But I think it's a fun system to study, and thinking about if you had a system, if you have a microbial community that doesn't have turnover due to, you know, things being washed away or something that's not a flow through system, this is a fun little system to use for that.
But I'm happy that they published this. Like, they didn't find what they were looking for, and they don't have a simple answer. And I feel like the papers that get published are unbelievably simplistic in what their findings. So I love seeing environmental papers where they're like, we can explain 3% of the variability. Like there's a lot out there that we have to keep looking at it. And if we only publish papers that we have, have our 3-line, here's exactly how this system is set up and controlled and organized, then we all lose out, because there is a lot of money being spent on these large sequencing projects, and they're not easy to explain, but I still think they're useful to do, especially if you can publish it as we tried a lot of things, we still don't know. And I'm very encouraged as a microbial ecologist that this got published and it wasn't rejected as you didn't find anything. Yeah, I know we didn't find anything because it's a hard thing to find. So I liked it.
Well, I think those are all very good summary statements. This is a good, I can't remember who suggested it. Was it Nicole? Who suggested this one? It wasn't me. I can't remember if it was Nicole or Kendra, but thanks to whoever suggested this. It was a good paper. So the journal club itself is going to go to a book club for the summer. So they've done this twice now, so this will be our 3rd time. So in June, July, and August, we'll be doing book club discussions rather than paper discussions, with the hope, perhaps optimistically, that the summer affords people a little more time for some reading books, which is sometimes true and sometimes not. This summer, what we're doing is the summer of genomics. So our theme will be genomics and genetics. We've got 5 really great book suggestions that are on the website. So if you go to the g+microjc.wordpress.com, which the link is actually in the YouTube video, so in the description. If you go to the website right now, we are running a poll on the Summer of Genomics page that you can vote for the 3 books or the book that you would like that we could make one of the 3. Right now in the lead is Fatal Invention, which is awesome. I've read it and I'd be very happy to read it again. It's in the lead, but then we've got 3 tied with the same number of votes. Like, I think right now each of them has 1 vote. So we need to get some separation in some of those. So if people haven't voted and want to go vote, please do. And then I'll be kind of trying to ask folks who participated in the journal club and people who are interested about when might be good times to do the summer book club. So we'll be anticipating that. And the first one will be sometime in June. So it gives people a chance to get the book and read the book. Any questions about that?
Where was the voting again?
Yes, it is. So if you're watching this video on YouTube, you can go to the description in the video, or the description on the video page. I've got a link to the Journal Club's website, and there's a tab that says Summer of Genomics. You can click on that. That gets you to a description of the book club itself, and then the poll is on the bottom. It's also g+microjc.wordpress.com. You can click on the Summer of Genomics tab. So hopefully everybody's able to join us for that. I'm really excited about the summer's book selections. They all look great. So I'm be announcing kind of the 3 that we're going with once we get some votes in, and then we'll be working on days and times. So in the meantime, thanks everybody. This was a really great discussion, and it's really nice to see everybody. Hopefully everybody has a good end of semester, and the next time we get together, it'll be for June for our book club discussion. So thanks very much, and we'll see you all later.